So thanks, everybody. We can go ahead and get started. For the folks online, are having some adventures with the AV, but I think we've got it figured out. So thanks, everybody. Thanks for your patience. Sorry for starting a couple minutes late. Really excited for our speaker tonight, Joanna Slusky. She's a professor at University of Kansas. Has been in the Rosetta community for many years now, back in the dark days before artificial intelligence, all the way up through, and really excited to see her work. Now moving into the AI and ML space. Her lab has been focused on membrane proteins, in particular the outer membrane proteins for bacteria, and she's made seminal advancements in this field. And so, yeah, I'll shut up and give the stage to her. So, Joanna, thanks so much for being with us tonight.
Great. Thank you so much, Chris, for the lovely introduction, and also to you and to Nick and Sergey for the really helpful audio-visual setup, and just for having this amazing venue where people can come and speak about protein design. I've been hearing for years about Boston protein design, and so when I was in Boston, it seemed like just a phenomenal opportunity, and I'm really glad you guys are having me. So thank you to you, and thank you to the audience for all hopefully being somewhat interested in protein design. Today, I'll be talking about evolution and design of outer membrane proteins. I said I was going to be talking about LSTMs. I will be talking about LSTMs, but it's going to probably come at least like half an hour into the talk or so, and then we should have a good 15-20 minute talk about those as well. But I thought that there were things that you needed to know about outer membrane proteins to make the LSTM part cohesive. So we'll do it that way. When I say that I'm talking about evolution and design, that's because I think of evolution as a way in which nature designed proteins, and we want to learn those methodologies to be able to use them. And when I say that I'm working with outer membrane proteins, I'm talking about proteins like you see here, and we see ourselves as ultimately playing cat's cradle with those proteins, which is what this is an image of. So I like to start with an aphorism. A lot of people like to say, it's not what's on the outside that counts, it's what's on the inside that counts. And that's true. But in my lab, we like to say, it's when your slide advancer works. That's important. It's what's on the outside that counts. Okay, maybe a slow reveal on that one, but the idea here is not that we're particularly materialistic or decamping, but rather that we look at the outside of a gram-negative bacterium. Here you're seeing a lovely picture by David Goodsell, where this is E. coli, and we're looking over here and we say that is an amazing platform for protein design. So what do I mean by that? Well, fundamentally, that location is able to control the interaction between the cell and its environment. And when you can do that, you can do all kinds of interesting things. So some of the things that we're interested in my lab are dealing with antibiotic resistance. So this protein over here, actually, that one is, his job is to push antibiotics out of the cell. And so the antibiotics can't reach their targets. If you could plug that protein, if you could stop that protein from folding, then you might be able to lessen antibiotic resistance. Similarly, we're also interested in, hey, could we have enzymes at the cell surface that could degrade pollutants? If you have enzymes at the cell surface, they don't need to bring in substrates, they don't need to push out products. Instead, they're just able to function and do something that is potentially useful outside of themselves. And finally, if you put this kind of protein across a membrane and put a voltage across that membrane, you could change perhaps the radius of that protein so that you have different abilities to bind different types of heavy metals and ultimately detect that binding by a difference in voltage across the membrane. And so we see these as possible heavy metal biosensors as well. Now, the thing about these proteins, with all of these different functions, all the functions that one would need to interact with this environment, is that they only have one fold. So let me tell you what I mean by that. If you look at the proteins that are responsible for causing the cell to stick to things, adhesins, or you look at import or export proteins, these are the bread and butter of the outer membrane. They're bringing in nutrients, they're pushing out toxins. If you look at assembly proteins, so the outer membrane also has proteins that cause other outer membrane proteins to fold. This one we'll talk about a little bit later, so sort of keep it in the back of your mind. It's called BAM-A for the beta assembly machinery. And this guy folds all other outer membrane proteins. And this here, it's an enzyme. Okay, so all of these, if you look between the red lines and the blue lines, which are shown there because they're the border of the membrane, then you see that they're all beta barrels. And not just are they all beta barrels, but they're all up-down beta barrels. So they're all completely anti-parallel, up then down, then up, then down. And in fact, they're all right-handed. They're all tilted in the same direction. And then the next strand is contiguous, so that it's strand 1, next to strand 2, 3, 4, 5, 6, etc. The difference, though, is that there are changes in strand number. And I've highlighted that a little bit here by saying this is a 12-stranded barrel versus an 8-stranded barrel. And of course, that can give you a different diameter. And as you change that strand number, therefore, because you get a difference in diameter, that's linearly correlated to the circumference, which is actually what you're changing. You can coordinate different metals, as I was saying before. You can also make various different types of enzymes. 68% of proteins with barrel structures are enzymes. And that's fundamentally because when you have this kind of barrel, you can sequester substrate from solvent. So this is the kind of universe of outer membrane proteins that I'm going to be talking about today. And having only one fold for so many functions, I think brings up sort of why questions and how questions. And I'll talk just a little bit about like, why is there only one fold? And this is something that I think I can have a more extensive conversation if you wanted to ask questions afterwards. But for the purpose of this talk, look, secondary structure, alpha helices and beta sheets. Fundamentally, what that's doing is repeatedly satisfying backbone hydrogen bonding. And so in a membrane, it's really important to satisfy that hydrogen bonding because in solution, your other option is water, but there's no water in the membrane. So when you want to repeatedly satisfy hydrogen bonding, you're going to have secondary structure. And so as it happens, and this is again, what I think we can have a longer conversation about after the talk, is that in the plasma membrane or the inner membrane, we have alpha helices, and in the outer membrane, we have beta sheets. And not just do we have beta sheets, but if you think about the beta sheets, right, this is now a beta pleated sheet. And the hydrogen bonding, again, within the backbone are those pink dots. Okay, so the sheet goes along here, but this guy and this guy and this guy are not satisfied unless it wraps around to make a barrel. And the other thing is that where your side chains are now is not along the line where you're doing the hydrogen bonding. So what you'll see is side chains here are these yellow spheres, and you see they're pointed up, down, up, down, up, down as you go along the strand. And what that means is that your residues in the beta barrel are pointed in, out, in, out, in, out, so that beta strands in the membrane tend to have this property where you get all hydrophilic facing in and all hydrophobic facing out. It's the opposite. It's hydrophobic, hydrophilic facing in, hydrophobic facing out. And so you go hydrophilic, hydrophobic, hydrophilic, hydrophobic down your strand. And that'll come up again later, especially when we're talking about what are the properties in generation and seeing through the LSTM. Okay, so now we'll talk about how. How did nature evolve this one structure to have so many functions? And as I said, my interest in this how comes from this idea that maybe we could use this ultimately for design. I do want to let you know, somebody has asked me, I thought this was a cool picture, we commissioned it, and someone said, oh, wasn't it cool that Cro-Magnon man was able to intuit protein structure? So right, that's not what happened. I just want to say this is from Batika Saxena, and it is not actually Cro-Magnon cave. But how did nature evolve this one structure to have so many functions? And a lot of people think, and this is, I think, both a simplistic and a somewhat complex answer, is that the answer is that these beta barrels are repeat proteins. Okay, so I don't know how many of you are familiar with repeat proteins, but the way that repeat proteins function is that you have the same domain, bless you, over and over and over again. And So if beta barrels are repeat proteins, then we know already that gram-negative beta barrels have an even number of strands, 8 through 36. And so you could have a hairpin, you could have a strand, loop, strand, loop, strand, 2 hairpins. Either way, what we're thinking about here is some sort of genetic element that gets duplicated so that ultimately you get this sort of topology. And I think it's sort of clear to see why this would be a repeat protein. But then if it is a repeat protein, then is that really the way that we get all diversity of this fold? And so we started looking at this, and the way we looked at this was with a network model. And we did this back in the dark days of science before AlphaFold. And so what you're seeing here are structurally characterized experimentally proteins. Each of those are in a colored box. And those colored boxes are structurally characterized membrane barrels, and an edge exists between the boxes if those proteins have a relationship with each other, and we can measure that relationship with a statistical tool called an e-value. Now we are doing crazy e-value over here because we are doing an e-value of less than 1, which means that there might be 1 false positive for every true positive. So not super reliable. You shouldn't believe all of these connections exist, but rather we're doing this because of the relationships that don't exist. That you see that lots of these proteins are not related to other barrels, and that's consistent with an idea that there was convergent evolution onto this fold. And I can talk about that more. This was work ultimately done by Megan Franklin, who now works in protein design in the Boston area. But for now, just know this is showing that there may be some convergent evolution on the fold. Most proteins, though, are related, and we call those prototypical barrels. Later, we might look at e-values that are more reasonable. And when I say that, what I'll say is like, this is an e-value of 10 to the minus 4. 10 to the minus 4 means that there is likely 1 false positive for every 10 to the 4 true positives. So that's where we are with e-values. Okay, and let's talk now here. These may have converged onto the fold, but the green probably diverged. And that's where we're interested in learning about this kind of copy and pasting duplication effort, okay? And so we do know that these copy and pasting duplication efforts occur. There are lots of ways to see it. One way to see it is if you think about protein alignments, oftentimes you think about an alignment between protein 1 and protein 2. But you can also do an alignment between protein 1 and itself. And when you do that, that's indication that there was some sort of duplication event that happened. And we can see it all over these structurally characterized beta barrels. I'm organizing them now by strand number. You get, as I said, even numbers of strands, 8, 10, 12, 14, etc., along the x-axis. And you can see some of them don't have it. Those are in blue, but many of them in red do have it. And so we say, okay, so there are internal repeats. That means we do have this duplication process occurring. And in fact, we believe that the primordial barrel was likely an oligomer. Now, this often happens for repeat proteins, where you have some unit. That unit can't function on its own, right? I told you that we really need to satisfy the hydrogen bonds on the edge. So this guy would not be happy inserting into the membrane. But if he made an oligomer, right, some sort of multi-chain barrel, then he could. This is a 12-stranded barrel. We got 3 of those. And then ultimately, maybe this was duplicated in such a way that these had loops between them, and it was one long chain, and that's how we got the first outer membrane barrel that was not an oligomer. Okay. So we do believe in internal repeats. We do believe that these duplication events happened, but we also think that other things happened, other things that surprised us. And when we were looking at some of the alignments over and over and over again, we would see alignments where on one protein, we would see a strand, loop strand, and the part that was the loop aligned with strands. And that was surprising to us because it's surprising when something that's outside of the membrane would align with something inside of the membrane. That's kind of weird. It was surprising to us because we thought that basically we were getting these strand, loop, strand, loop, strand because of these duplication events. And this would be some sort of diversification just based on mutations. But we did see it over and over again. We did see it for E values that were fairly safe, E values where we started to believe it. And so that made us think, hey, does repeat protein topology, can that be created without genetic duplication? And might that be a feature of the membrane enforcing this kind of fold, just like it seems to be enforcing this fold when it comes to convergent evolution? So we said, hey, can we engineer this? Can we do protein design? And can we make this 16-stranded barrel into an 18-stranded barrel using a loop to hairpin mechanism? And we did this in a fairly simplistic way, where we took one of these alignments. Again, this was a e-value of 10 to the minus 5, and this was poor B Neisseria gonorrhea along with the major outer-membrane protein of C. jejuni. And we took this part and we just inserted it here. And you might say, okay, maybe strand integration should be trivial. Like if you add a bunch of protein, shouldn't it just like make a strand of its strands? I think it's not that simple, and it's certainly not that simple in our hands or in our colleagues' hands. Integration of strands into OMP-G was unsuccessful, and that was published. Integration of extra strands into LAMB was also unsuccessful. And the one thing that ended up really working that we found in the literature was that using evolutionary methods is really helpful. So what was shown was that if you duplicate an 8-stranded barrel, you can make it 16-stranded barrel. Again, this is the kind of evolutionary duplication that we think existed in nature. And so with these unsuccesses and the fact that unsuccesses are generally not published, we weren't sure what to expect. And so we started with AlphaFold, and this is work now that was done by Rick Dar, who also is working in the Boston area. So Rick first looked at AlphaFold, and he said, what happens when I make this, what I'm hoping is an 18-stranded barrel? And AlphaFold gave 2 answers. It said it could be an 18-stranded barrel. And in general, these guys tend to be trimers. So you see it here as a trimer. It could be an 18-stranded barrel. And in that case, here's the strand, kind of low, probably low confidence on that one, but it's over here. And that means that you've got less in the lumen of the core. Or it could be a 16-stranded barrel where you started, where you've got more confident strands, but less confident loops, lots of loops in the lumen. And so we weren't sure. And so then we made it and we tested it and we did some CD. And so here what you're seeing, poor B wild type is that 16-stranded barrel, the native protein. And poor B18 is our hopeful name for the protein where we added a bunch of amino acids and hope that they make strands. And so what you're seeing is that you're getting more beta sheet conformation in poor B18. So that's hopeful. And in fact, if you model that now and you say, okay, here, anything with these hash marks, that's from a PDB to CD, where you go from a structure to a potential PDB signal, and then you can put this into here to get the beta sheet amount. And so you've got the beta sheet amount on the y-axis. Poor B wild type matches up pretty well with what you would expect from the PDB to CD. And if you compare what we actually got here in blue to the possible structures, what you see is that it's closer to the 18-stranded model. So that was good. And then we went on to do some single channel conductance. And what you see is that you get more conductance for poor B18 than from poor B wild type. And if it were 16-stranded, you would actually expect it to get a lot less conductance because of all that crap in the lumen. So it's statistically significant, the difference. If you model the difference of what's going through, then you would expect, again, poor B wild type to be slightly smaller than poor B18 if it's 18-stranded, but poor B18 if it's 16-stranded would be super small. And so you can actually take the conductance and the potential radii and match them up. And this is where that conductance should be if poor B were 16-stranded. And where it actually is very close to it where it should be. So we said, okay, there are frequent duplication events, but genetic duplication is not required for repeat protein topology. The membrane seems to really be enforcing this beta structure, and it seems to be willing to acquire in this very weird particular way. Loop hairpin evolution, therefore, can be engineered. So I said, okay, but what if we knew a lot more about beta barrels in general? Then could we know even more about evolutionary mechanisms and folding mechanisms? And so this is how did we get the database of lots and lots of barrels? And that's what we're going to then use for the LSTM. So we're getting a little bit closer if you only came here for LSTMs. Okay, so database creation. What we used here was multiple sequence alignments. Many people here, maybe even most people here, are familiar with multiple sequence alignments. If you're not, I made a helpful picture where instead of using 1,000 proteins, I'm using 4, and instead of using amino acid letters, I'm using widgets. This is a basic multiple sequence alignment, and the idea with sequence coevolution and coevolutionary contact maps is that there are positions that are varying with each other at similar rates, and that means that they're likely near each other in space. So from that, you can get contact maps like this. And the way you read a contact map is that the y-axis and the x-axis are both amino acids, the same amino acids. So you're going for the same protein from 0 to 300 or from 0 to 300. So this is the self-interaction. Darker is higher contact probability. And the thing about these beta barrels, and I told you that all of these membrane proteins are beta barrels, they have these super consistent contact maps, where you can see here, this is strand 1 interacting with strand 2, and this is strand 2 interacting with strand 3, and this is strand 3 interacting with strand 4, and you keep going until you get to the first strand from over here, interacting with the last strand from over here, and that is the closing contact. So these are the kind of maps we expect to see, and knowing what we expect to see from that ended up meaning that from sequences, we could get these multiple sequence alignments and identify if something was an outer membrane beta barrel based on these contact maps. And the person who did this algorithm was Rebecca Bernstein, who at the time was a high school student in my lab. And it's a fairly elegant algorithm where she said, okay, is there a closing contact? That's where the first strand interacts with the last strand. If yes, it's a transmembrane beta barrel. Otherwise, you can count the number of hairpins with particular lengths. If you get enough of them, it's a transmembrane beta barrel. And this alone was able to give us a balanced accuracy of about 96% and extremely low false positive rate. So what does that exactly look like, and how did we measure it? We looked at a data set where we had 121 outer membrane proteins. These were the structurally solved ones, not so many of them. And we compared it to 1,000 non-outer membrane proteins. Now, when you have a sort of asymmetric data set like this, the problem, of course, is that you have an algorithm that says nothing is an outer membrane protein, then you get like 90% accuracy, but this doesn't really tell you anything. So we look at something called the Matthews correlation coefficient, where you're looking at the true positive and the true negative and the false positive and false negative to get an overall truthiness score. We can also look at the individual true negatives, false positives, true positives, etc. And we call our algorithm, is it a barrel? And we do quite well. So here I'm comparing it to other sequence methods of telling something's a barrel, and I'll tell you more about these as we test our LSTM, so they'll come up again later. But what you notice is, number one, we've got the best MCC, but also we have that good MCC because we have very, very few false positives. And that's really good, because if you want to really believe your data set, if you want to be sure that everything in your data set are really barrels, then this is the way to go. So we have this algorithm, is it a barrel? And we start screening through all kinds of proteomes. So one question that we can start asking is, how many outer membrane proteins per organism? And so this is all work by Daniel Montesano, a postdoc in my lab. And he started at looking at 3,000 reference proteomes. He would take the barrels from previous data sets. There were a lot of false positives in those, so he removed the non-barrels. And then we would identify what phyla or class they belong to. You can see we're doing all kinds of bacteria. We've got Bacteroides over here, Firmicutes over here. And then you would search those proteomes, and for particular contact maps that looked like the ones we wanted. And what we ended up getting is now in color, so you can see the difference between what was previously known and what we have now, where if you look on this rightward axis, you can see the fold increase where the asterisks are. But overall, you can see that there are like, lots of different types of transmembrane beta barrels in Bacteroidetes per organism. There's somewhere on average around 140, but very few for the firmicutes. Bacteroidetes have the most, right? And then actually gamma proteobacteria is the next. Those are the kind that are most commonly studied in labs. Firmicutes have the least, but we're getting 50 times more than previously predicted. So lots and lots of beta barrels. And then we went even further, expanding the data set. And we figured out a trick that let us expand the data set quite a bit. And the trick was that we started with 30,000 sequences. Some were barrels, some were not. And we compared all sequences against all sequences and recorded if they were both barrels or not barrels, and how related they were. What was their sequence similarity? So we did pairwise sequence similarity on the x-axis, and if they were both barrels or if they were both not barrels on the y-axis. And what you're seeing is that there's really no correlation until you get to about 40%. And at 40% sequence similarity, proteins 98% of the time have either both barrel or both not barrel assignments. So we could say if something is 40% sequence similar, then we could just assign something that we know as a barrel or not barrel and say that that's a barrel too. So that's the trick that we used. Given that it's 98% in terms of that probability, that's a higher confidence than we had through the algorithm to start with. So we felt pretty confident with it. And that led to us having 1.9 million outer membrane protein sequences. It's an extremely large data set with high confidence. And so what could we do with that? So we started by saying, okay, what are the types of outer membrane proteins? Before I said, I think there's some convergent evolution that's happening. If we look at what's related to each other, we ended up getting 11 groups out of the 1.9 million. And 6 of those were never crystallized. So here are the prototypicals. That's the one I was talking about before. Most of them are there. We've got 1.9 million, 1.5 are there. But we have all these other types, Tim Usher's and LPTD-like. Those have all been structurally resolved. But we also have things like TSX-like, where, NFRA-like, where it's been annotated in uniprot, but not structurally resolved ever, except for AlphaFold, of course. And then, then there are things that had not even been given names. We've got domain of unknown function 1302, where we've got 5,732 examples of proteins that seem to be beta barrels and had not been typed. We've got hypothetical protein type 1 and hypothetical protein type 2. So we have these 3 types that have never been annotated. So lots to look into, lots to explore. Now, the next thing we wanted to do was be able to see if there were these duplication events in barrels. And we wanted to investigate the length of these proteins in order to see those duplication events. But the length isn't fully representative of whether or not that's part of the barrel, because there can be other domains. So we looked for the closing contact. Now, about 25% of our proteins had a closing contact. And when they did, we could map that so that in terms of the structurally resolved ones, we could compare the PDV barrel size to when we found the closing contact, and we got a pretty good score in terms of R squared for whether or not there was a relationship between the barrel size and what we were predicting. So then we looked at the lengths of these proteins, of just the barrels, I should say. And for a lot of them, there was just a single node. Okay, so that you're not seeing a lot of evolutionary duplication for that group. But for the other parts, you are seeing evolutionary duplication. And in fact, I would argue that you're really seeing like duplication, because 161 is about half of 333, and 242 is exactly half of 44, and 232 is exactly half of 464. So, so we're seeing these duplication events that I was telling you about before, all throughout nature, which is pretty cool. Now, another thing we were able to see was the beta signal. So you've probably heard about signal sequences for like targeting the membrane in terms of signal peptide. Beta barrels have to go through the inner membrane translocon and then to the outer membrane. And so they have the signal peptide at the N terminus. But at the C-terminus, they also have something of a known sequence motif. And that sequence motif has a complex possible name where they say, okay, so it could be that you've got any polar residue and then something, and then glycine, and then something, something, and then an aromatic residue or hydrophobic residue, something, aromatic residue, hydrophobic residue. But that's the signal sequence, and that in the client protein that's getting inserted into the outer membrane is at the C-terminus, like I said. So what you're seeing now, and I told you about this beta assembly machinery at the beginning. This is the assembly protein that's pushing things into the membrane. That's in red. Sorry, no, that's in blue. That's in blue, and in red is the client protein. And these guys join together with the client protein's beta signal. So that's what you're seeing right there in green. That binds to BAM-A, and that helps it insert. So we looked for C-terminal sequence motifs for all 11 groups. And what we found was that they all have some sort of sequence signal at the end, at the C-terminus. Sorry if I said that. C-terminus. We looked at the C-terminus. We found it at the C-terminus. And all of them had sequence motifs, but only the prototypical barrels had the classic one. Okay, all the others had really different other ones. And that made us think, okay, so these large differences among the motifs might mean that there are more ways of insertion than we currently understand. And that those other ways of insertion might be with BAM-A or not with BAM-A. And that's something that we're interested in investigating further. Now, we also looked at all kinds of different bacteria. So now this is by phyla. So really, really, really diverse. And when you see this, you can see that the 11 different types are kind of checkerboarded. Now this, we're trying to organize this by relationship, although bacterial relationship is somewhat contentious within the field, but we did our best and we don't claim to be experts on this. But what you'll see is that for the prototypicals, and prototypicals are in all of these, for the prototypicals, you see this really common sequence motif, and for most of them, all through the, certainly all through the grassophytes as well as part of the Pterobacteria, the answer is yes. So you can see those here. 20 of the 34 phylos prototypical proteins have a very traditional beta signal in the prototypical barrels. And the consensus for that, we would say, is LGLGYRF. So you can see LGLGYRF over and over and over again. Sometimes L is a little bit more preferred, but ultimately, out of 20, this is the probability that you're getting any given one of those numbers. So it's working out really well. So these similar sequence motifs indicate that although we saw before that there's sort of all different sequence motifs, meaning that they're all possibly being inserted in different ways or not with BAM or things like that, that all of these organisms probably are using BAM, but just maybe not for those other proteins. Okay, so I saw this picture on Twitter and it said, truckload of beta sheets going up to M6. And I was like, that's what I have. I have a truckload of beta sheets. So we have a data set of 1.9 million bacterial outer membrane proteins. It's the largest data set and most accurate way of identifying outer membrane proteins. And we have it so that you can view it at your leisure and search through it at isitabarrel.ku.edu. Now, 3.5% or less of bacterial proteomes are outer membrane proteins, and that depends on class. And 11 types of outer membrane proteins seem to exist and seem to have convergently evolved onto that fold. Some of them may be related to each other. We haven't found that relationship yet. 3 of those types aren't even previously annotated. And the barrels show lengths consistent with duplication events. So that is consistent. And the beta signals show possible diversity of insertion mechanisms, while the conservation of the prototypical beta signal supports conserved use of the BAM translocon. So now we have a data set to train on, and we can start talking about training and machine learning. So can we use this for barrel generation? And I'll start with that part of this talk by saying membrane barrels are considered a challenging design target overall. So there's some landmark work from Anastasia Bordevia, who was at the time in the Baker lab and now has her own lab. And at the time, she designed parametrically beta barrels de novo for the membrane. And then more recently was able to better control the pour of the de novo design barrels. Again, I believe that design was also parametric. And throughout that work, one of the standout scientific facts, I think, was that there were difficulties, but also opportunities with using glycine kinks. But so far, to my knowledge, there haven't really been any generated beta barrels for the membrane. And so we are trying with an LSTM, which is a weird thing to do. So an LSTM is a long short-term memory model. It's small, okay? So it's so small that we're using about 100,000, 200,000 trainable parameters. We're using 400,000 sequences. This would be in comparison to trainable parameters of between 100 million to 15 billion or something like that, and 500 million sequences. So at every step, we're sort of 1,000th of the size of the large models that you're probably used to looking at. And we're doing this in collaboration with two computer scientists, Rachel Kolodny and Rita Asadchi, and both of them say, like, you just have to understand that it's like not a legitimate choice to use an LSTM. So okay, it's not a legitimate choice, but they're still working with us. So we'll move on. And one of the things that we thought we could ask was, how much power do you really need to generate a novel protein? In Looney Tunes, there are these things called gremlins, and this gremlin has a comically large mallet for the problem that he's trying to deal with. Are we just, at this point, gremlins with comically large mallets? And so this is how this LSTM works. And again, this is more work by Daniel Montesano in my lab, where for the training, we start with pieces of our sequences and we input those pieces of sequences. And what we get as we train from the LSTM model is a propensity of all the different amino acids for being next after we've given it 50 amino acids. And we can compare what it thinks is best at next with the actual thing that's next. And we can then use that loss to update the parameters and keep going over and over and over again in what are called training epochs. That would be the time measurement. And we can look at like a loss measurement. And here we're looking, and this will come up a bit later, with a bunch of different learning rates. So you can have faster learning rates. That would be like 5 times 10 to the minus 4, or slower learning rates, 5 times 10 to the negative 6. And we can also use a scheduler. That's what's shown in pink. And that means it's sort of like simulated annealing, where you start out fast and then you get slow as you go along. So the scheduler ended up being best. And when we do generation now, what we do is we can just sort of start generating amino acids, saying to the LSTM, what do you think is next, even if it doesn't have that context? And then every time it makes something, it's making its own context. Then keep putting residue by residue, another amino acid. What do you think is next? What do you think is next? Okay. So are we making barrels? I don't know, maybe. We're not sure, but I'll tell you what we do know. So what is being generated? We know that we're getting pretty good amino acid distribution, pretty similar to beta barrel. So if you look at a beta barrel amino acid distribution, and now we're looking epochs. On the x-axis, I told you this is sort of training time. On the y-axis, you've got all the different amino acids. The first generation, it doesn't know what's what, but immediately after, it's able to give you approximately the right distribution. So this is delta median abundance of color, and blue means it's the same, and red would mean it's totally different. And basically, it doesn't get cysteine so right, but other than that, it's really pretty close. Bless you. And we can look for sequence, beta signal sequence moments. So I told you before we had this beta signal. We sort of use an approximation here of the beta signal as GXXXXF. Not all beta barrels have exactly that. 40% of the structurally resolved ones did, and we're getting less than that, but not none. Okay, and we can also look at how novel it is. So I'm going to go back to this concept of e-value. Up here, which would be an e-value of 1, 10 to the 0, we're seeing less likely to be related. And down here, 10 to the minus 4 would be more likely to be related. And we're seeing that most things are pretty much up there. So lots and lots of novel sequences. Now we can also look at the metrics that I was telling you about before when I was comparing how does is it a barrel do compared to these other sequence metrics. So there are these sequence-based barrel features that can be determined by programs like PredTMBB2. And so again, epochs on the y-axis, color based on like, no, 0 is 0 and 25, 0.25 is up there in terms of density, so that's more. And the way I'm going to compare these things is we're going to look at solved beta barrels. So that's this line right here, and you can see that PredTMBB2 really predicts solved to always be yes, one is right, they're beta barrels. Shuffled means that we're taking those solved beta barrels and we're shuffling the sequences. And so PredTMBB2 actually thinks that lots of the shuffled sequences are in fact barrels, and that's because basically it's the way they're looking at the sequence. This amino acid distribution, and random is random amino acids, and that nobody predicts to be like beta barrels, but it's a control. So when we look at PredTMBB2, the way that works is that it broadly considers sequence composition and amino acids in context using an HMF. So the fact that you generally get the amino acid distribution right means that even shuffled sequences are doing quite well, although the generative LSTM that we're using does slightly better. If we look at this Freeman-Milne beta barrel score, the way that one works is that it's taking into account both overall amino acid distribution as well as hyperphobicity alternation. Remember I told you with the beta strands, you're going hydrophobic, hydrophilic, hydrophobic, hydrophilic. It's taking that into account where it says, okay, an average strand is about 10 amino acids, and you probably get a loop that's about 5 amino acids, and another strand is 10 amino acids. So it's getting that sort of hierarchical topology that way. So now you don't see the same density in the shuffled, but it's still scoring our generated designs fairly well. And here you can get bump where that's like the Freeman-Milne, but with context-free grammar. And also it has a number of features like the beta signal, where it says, is it a barrel? And instead of being sort of from 0 to 1, like you saw before, it has 5 possible answers. 0 means it's not a barrel. And then 1 means it has some features of the beta barrel. 2 means it has more. 3, it has even more. 4, it has even more. 5, it has the most. And so what you see is that even beta barrels themselves don't always get put in 5. So if you're looking at the solved, you get a bunch in 1, you get a bunch in 4, you get a bunch in 5. Shuffled does worse than that. We're doing better than shuffled. We're getting more in 1, more in 3, but we're not doing overall so great. And so then I wanted to look at these metrics just a little bit closer because I think there's something that we can learn from them. And I wanted to sort of just take a moment to say that I do think in general there's a lot to learn from machine learning, and that it doesn't all have to be a black box, or at least a black box in the way we talk about it, that a lot of machine learning coming from the field of engineering ends up being like, well, what does it make? Can we make it better? But not how is it making it, or what's really going on, that we have this image of a black box where you put an input and you get out output, and better is better, and that's if it's better, then we're just going to keep going for better. But there's another type of black box, which is the black box of an airplane. And we specifically look at the black box of an airplane to tell us what's actually going on. And so I do want to encourage us to be trying to figure out what's actually going on. And here, ooh, that was fun. If we slow it down, so this was all with the scheduler. I hate when that happens. Okay. Ooh. Okay. Here's with the scheduler now. Here's like a 10 to the 6 minus 5 times 10 to the 6 learning rate. So this was more comparable to 5 times 10 to the minus 4. So we slowed it down 100-fold. And we can start to see, like, oh, at around the 30th epoch of 5 times 10 to the 6, it started to learn PredTMBB, which again, we said was amino acid composition. Whereas we see that transition actually, that's slower, I should say. You can't get to it. You have to use the 5 times 10 to the minus 5 learning rate So this is the slower one. These are faster, where you have to use a faster learning rate so that it's happening slower. Okay, right? If it's happening slower, then you need to see it over a faster learning rate. If a faster learning rate is being used, then you're seeing even more things, both the slow and the fast. Whereas if you're using a slower learning rate, you're only seeing the slow things. Only seeing the fast things because they happen over slow time. Okay, so these are happening at sort of 10 to the minus 5, where you're getting what you would have otherwise here, for better or worse. So what that says then, right, is that for the overall amino acid distribution, you're learning that faster, and local amino acid patterns for secondary structure, you're learning slower, and even slower would be perfect sequence motif recapitulation. That is quite hard. But all of this is in bulk, right? Nobody actually expects every sequence generated to be perfect. There are ways to filter and get the ones that have all the features you want. And will those actually be barrels? And we didn't know how to do that other than to test it until this paper was published this summer. And I'm very grateful for it. It's also from Anastasia Verbeda's lab, but it also has Sergey as a co-author, so thank you. But the idea here, this was the figure that we were really excited about, because, okay, so because they measured the ESM folds mean PLDTs of all the proteins that they had designed in that first science paper where they designed transmembrane beta barrels. And I'm gonna just check my connections. Oh, that was first. Okay. And what they found was that if you're looking at no expression in this jade color and folded, no expression or misfolded in the jade color or folded in the gold color, then there seemed to be this line where at around 75 mean PLDT, everything was folded. And we said, oh, well, we could measure by ESM fold the mean PLDT. And what we found was that by that metric, at least, our LSTM was generating barrels. So if we have 250 sequences generated, we got 5 barrels with an average PLDT of higher than 75%. So that's 2%, but I don't know how much, I mean, filtering is filtering, and the computational time that this takes is completely negligible. So here are some of the barrels that we found. I can tell you a little bit about it. This one's 22 stranded. It does have something kind of like a beta signal, but maybe it's only 4 to 3% beta signal or so. This one actually has a 23% identity match in blast. So it's sort of recapitulating possibly something it was trained on. Whereas this one is a 10-stranded barrel. This one really does have a nice strong beta signal at the end. And it has no blast relatives. Similarly, this 8-stranded barrel has something like a beta signal over here and no blast relatives. Here's another 8-stranded barrel. This one does have a beta signal for sure. No blast relatives. And finally, for the 5th one, we're seeing something maybe like a beta signal. Certainly has the GXXXX sequence, and that one is a homologous 30% sequence identical to a beta barrel. So what you're seeing here now, just to mention, right, is we're coloring it in alpha fold convention, where very high PLDT is dark blue and very low PLDT is orange. And so we were excited to see that these were coming just out of an LSTM, but it kind of made us wonder, like, why is this working? Because we gave something that we thought that was fairly weak a fairly hard job. And so I want to talk about that with you. Here are 5 answers. I think we can sort of mix and match these. More than one might be true, but perhaps membrane constraints make the barrel fold easier to generate. We talked about how we're converging onto this one fold. Maybe in some way, the fact that there's so much constraints preferring the beta barrel, maybe that makes it more likely to be able to generate it simply. It's possible that we have just a particularly good training set. So bacterial proteins diversify super quickly, right? They've got terrible proofreading, short generations. So we likely have better than usual evolutionary sampling. And maybe that's really helpful for this being able to generate fold. It's possible that this is a case where a small, well-maintained model can outperform a larger generalized model. And that's not the only case in the literature of such a thing existing. So perhaps that's it. But if that is it, then it might be the case that the generation method works for many folds. And that would be useful to know. I could just stand here or, and finally, maybe it's not working. Oh, okay.
I wonder
Thank
if it's
you.
just
What's going on? But maybe it's not actually working. So we haven't actually experimentally validated it yet. And we would know better if it's working if we would do that, and we'll get right on that. Okay, so just to sum up, and this is my last slide, so the fact that it's going badly isn't the worst thing in the world. But what's working so far? We have a small model, this LSTM. I said it's a thousandth of other models. It is generating membrane barrel-like sequences, and it has similar amino acid content, and many of the things that we generate have amino acid content, not just that's similar, but that has sort of topological representation where we can see hydrophobicity, alternation, strand topology, etc. We do see some beta signal sequence motifs, and many of the sequences are novel. And when we slow this down, when we slow down the learning rate, we can see that not all features are equally easy to learn. And in fact, it's much easier to learn the amino acid content than the specific motifs or the strand topology that we're seeing. And ultimately, some sequences have all the features and are novel and are predicted well by ESM code, and we're very interested in characterizing those. So with that, I'd like to thank the people who did the work. I talked a lot about Daniel Montesano, and I also talked about work done by Megan Franklin and Ryan Feehan and Rick Dar and Rebecca Bernstein. And this was work with collaborators, Rachel Kolodny and Margarita Sadji and Nirvan Tal, Sergey Gaina Mignacci and Matthew Copeland. I'd like to thank funding. This work that started through an NIH New Innovator Award and a Kay Embry. And ultimately, we are now working on it with NSF, Molecular Foundations of Biotechnology. And thank you so much for listening.
That was an awesome talk. Thank you so much. Actually, my first question is back for the first half of your talk with predicting the is it a barrel stuff. Super fascinating. And I think what particularly struck me was the potential for BAM independent insertion. And just would love to know a little bit more about your thoughts on that.
Yeah, so I mean, I think there may very well be other translocons. We know that like there's TAM, which is a homolog of BAM. That's probably not what we're talking about. There may be other translocons. There may be proteins that are translocon-free insertion mechanisms. You always have to wonder with that, and it's a central question in the field of outer membrane proteins, is that when we make in the lab outer membrane proteins, we can spontaneously insert them into mimetics, vesicles, things like that, in ways that you could never do for helical membrane proteins. And that's great, but we know that generally speaking, you need BAM, and it's required in bacteria. And the theory has always been, or maybe not always, but certainly I've been hearing many theories about that if you didn't require BAM, then when you went through the translocon in the inner membrane, what would stop the protein from inserting back into the inner membrane where it's closer, not having to get through the peptidoglycan, and ultimately being completely useless because it's not meant to be there. So there must be something that makes it difficult to insert into membranes generally, right? That then makes the BAM required. At the same time, what I showed was that we don't see a consistent signal sequence for LPTD. And certainly there have been structures or various information about stalled complexes with LPTD and BAM. So I think alternate ways of getting through BAM is the easy answer, but that there, biology contains multitudes, and there are probably other methods that I can't imagine right now. I'll do that.
Yeah, thanks for the talk. Are you able to tell if your models are adding sequences?
Repetitive sequences meaning like sticky, like G, or repetitive sequences meaning like it's just giving me the same proteins that I got out.
See, like domain, like your, like, how square generated. In other words, like if you were to do a repeat of the design sequence.
Oh, I see. Would I see internal repeats
Right.
in the proteins? Oh, I've never done that. That's a fun question. I don't know. I mean, can't predict, but it's a fascinating question. Yeah. I'd love to see internal repeats in my design proteins. Cool. Okay.
Yeah. Actually, I was wondering, so people usually hate on LSTMs because they practice have a hard time doing long range.
Yeah.
Do you have some sense of feel like that's a problem when you generate sequences, or do you think it's their?
We're trying to figure that out, and Daniel's working on that today. That it seems like with the short-term part of the LSTM, we're already past that at like 2 strands probably, and yet it keeps generating a protein. It can ultimately generate sort of a closing structure. It gets to the end and it's like, here's the place where I put my beta signal. That's all part of the long range thought of it. So we're trying to figure out like, what are the short range limitations of that? Question for you?
Yeah, it looked like some of your generated structures had 2 bright yellow.
Yeah. Okay, good thought. I didn't highlight that, but that's absolutely the case. That is the closing. That's the closing contact. Those are that's the close right there is, and you can sort of see it because I pointed all of these in the same direction. So here is the N terminus, and here's the C terminus, and that's the closing over there, and that's the weak link. That's always the weakness for us.
Do you think it's
Related to the question of Sergey? Look, Sergey's question is a very good one, and the answer is I don't know, but we're looking into it, and our computer science colleagues, Rachel and Rita, are particularly interested in exactly that question of like, okay, if it's working, how is it dealing with sort of when does the long term stop and when does the short term stop and things like that. So yeah, I think it's hard. It's hard to go all the way around, and I don't have an answer other than that. Other than we see a lot more barrels than this where it doesn't quite close, and so these are the ones that are predicted to actually get to closure. But we're lucky when they've got some confidence over here like that, whereas generally it's more like that.
When you first introduced the LSTM, you mentioned 50 residues.
Yep. As context.
Does it all, when you start generating a sequence, do you have to give it 50?
No,
Does
when we
it
start,
generate
we start at nothing.
does it generate a end?
It doesn't start with it. It generates an end token,
Generates an end
Yes.
token.
but it doesn't always start with M.
I see. I was just curious about it. It almost seemed like it provided 50 neurons.
Yeah, no. And so once we start generating, we can start it.
Very nice talk, thank you. And so given that there are similarities in sequences across these outer membrane protein beta barrels, what are your thoughts on outer membrane proteins and gram-negative bacteria as
I mean, I selfishly, as a person who works on them, hope that they're great drug targets, right? These are bacterial proteins. We find that many of them, so with drug targets, what you're looking for is something that's different in what you're targeting than in you, right? And so it's great that bacteria have this outer membrane and our cells don't, except the mitochondria. And so there are some of these beta barrels that are more related to mitochondrial proteins, and there are some that are less. So if I had to guess, not being a drug designer, I would be like, oh, well, I'd go for the ones that are less related to mitochondrial ones. And certainly within the context of that convergent evolution, we see actually really big structural differences, subtle, but but noticeable structural differences, maybe I should say, between efflux pump proteins. Actually, the antibiotic efflux pumps are structurally quite different. They tilt differently than the other proteins, and so I think that those might make better drug targets long term. And we're seeing that BAM has become a great antibiotic drug target with dargavectin. So I'm hopeful that there is life to that. Is your question?
Specificity against valuable human symbionts,
Correct.
that's an issue. Was that, antibiotic that will kill the infection without bothering,
Right. So that's why I'm
Human
saying, like,
symbionts.
it's a good thing that humans don't have outer membranes,
but our symbionts.
but our
that we
symbionts do. Oh, so sorry. You mean like how do we protect our microbiome, or how do we protect our
Yes.
mitochondria?
Yes.
Microbiome. Yeah. So then you would have to get much more specific. And look, there are, as we were saying, this whole range of proteins and slight differences in BAM and things like that. And something we look at is when we make a peptide that binds to an outer membrane protein, can we make it just bind to E. coli and not Pseudomonas, or things like that, where they have similar proteins but not identical? It's something that we look into, and I'm not as sophisticated a drug maker to know if I'm even close to getting that right.
Thanks for taking up. I didn't miss it. Did you also, what was the pattern sequence, the long sequence that you did in training? Was that also 50? Anybody say?
The training was regions of 50. Yes.
I think for the long, the LSTM 50 is really short, but anyway.
Okay.
And also with the loss function that.
How long do you think it's long?
I have done with the text model, you can go up to hundreds.
Hundreds of tokens.
Yes, but it is a case for a careful design. And here, we don't have that much diversity, we only have 20.
Right, 20, 21.
And it's not like before. But one thing that LSTM can do is when you train the model, you can pass the same data that you train and also do a clustering downstream. Maybe you have done what
I haven't.
Yeah.
Say more.
Because when you do the clustering, you already know the sequences, and you know the group, and you know which one should go to which one in the clustering, right? And then afterward, maybe you can, when you generate the sequences, then you mix the generated with the trained data and do pass it through the model again and do the training and do the clustering. Sorry.
Does it improve? I mean, a lot of what if you say garbage? Give it back.
Then at least it groups and make clusters that the generated sequences,
Oh, I see.
they should go to
Oh, I see.
a similar groups
Cool.
in the
We can
nature,
try that for sure.
right?
Yeah, I see.
And then LSTM has another feature which is called bidirectional.
Yes, this is a bidirectional.
This is bidirectional.
Yes.
Okay. Yeah. Cool or
Quick question, you're kind of skirting around
Yeah.
this a little bit. But why, when we went to eukaryotic cells and went to single membrane, because the variable is common when you're double membrane and you're during this interval, why was this fold lost when you're going into eukaryotics and you're getting into more alpha helical structure, but you're not seeing the beta barrel in single membrane?
So I can't necessarily say why, but I have a hypothesis of how. I don't think it was lost because we still have it in our mitochondria. But I think it's fundamentally not, I think that the reason that we get helices in our inner membrane and then barrels in the outer membrane is really a biological function of the sec translocon. That the sec translocon has a lateral gate that opens and allows for a helix to partition into the membrane. And if that helix is sufficiently hydrophobic, then it will partition into the membrane. So membrane helices are super hydrophobic. That's how you tell what they are and things like that. I just told you that the beta barrel strands are alternating hydrophobicity. Hydrophilic, hydrophobic, hydrophilic, hydrophobic. Ultimately, they have similar hydrophobicities to soluble proteins that are just inside out. So they pass through the sec translocon, okay, because they don't partition into the membrane. So that allows them then to go forward. And so when you have a single membrane, then what goes into that is what's partitioning into the membrane via the second transmembrane. Sergey,
I guess a comment and then two questions from
please.
online.
Okay.
I kind of wonder if this slide kind of answers the questions we were asking earlier, because it looks like at 8 strands, you don't see any of the yellow.
Yeah.
And as soon as you hit 10, you see a little bit of yellow.
So you
So I
think that's the
wonder-
border?
I wonder if you're cut off for long range, essentially like around 9 or something like that.
Could be. Yeah.
Might
I
be interesting
just
to like
noticed
get the PLDTs and just plot them for the NC terms and see like
Yeah.
what's number of strands that is something like starts to drop.
Yeah, we have a paradigm, I hope it works, but maybe we'll do your way, where we figured we would sort of mask and then generate some amount, and depending on how much we're masking and how much we're generating, see sort of where that line is of it not working out anymore. But
Right, so
we'll try.
question
Yeah.
from JP. I wonder if the LSTM be run over a sequence to generate a likelihood.
Say it again. I wonder.
I think they're wondering if you can use an LSTM to compute a likelihood for a given sequence.
Get a likelihood for a given sequence. I mean, I see. So, like an adversarial network, or
I think they're
Yeah.
probably wondering, like, I mean, I guess the answer is probably related to your loss function.
right.
I mean,
Okay.
supposedly, if you're giving it sequences, you could probably score them somehow.
Yeah. I don't know how to score with my LSTM, but I'm not opposed to it. I just don't know how to do it. Yeah.
And another question from Rakesh is, have you tried fine-tuning a protein language model on your data set and then generate sequences and make comparisons to your LSTM model?
We have tried fine tuning and doing other various sort of foundation model techniques, but we haven't had those do generative design yet, but we could compare them. That's something that's being more formally done in the Osachi and Kolodny labs. Yeah, good question.
How well does the construction model generally perform? There's a unique size with whether you have benchmark ESF or helpful to whatever the model.
Right.
you're using.
I didn't, but Anastasia did, and Sergey may or may not have been involved in that. I don't remember, but Anastasia showed that AlphaFold and ESFfold is really well on data sets. There's also the third sense.
I'm also really curious that like if you have those predictive structures narrow, does it perform similar?
So it's a good question. If you don't have any blast homologs, then you can't make an MSA. And you can't make an MSA, then you can't get that coevolution map, and then you can't put anything into it. Because the fundamental input is in a barrel. And so we could do it with the ones that have blast matches, but it's blast matches to an outer membrane protein, and so it would basically all be picking up outer membrane proteins, and so it would like be shocking if it doesn't see it is in barrel. Yeah.
My question's related to, is it a barrel? And the proteome searches that you did. You had this nice plot where you found in your 3,000 reference proteomes that you were searching, essentially a bar chart of how many different outer membrane proteins you found.
Yeah.
And some had many, and
Yes.
some had very few.
Yes.
And I'm just wondering if there's something that explains that. Is it like, the higher order organ, is it more complex organisms? Is it something related to phylogeny?
Right. So
What might explain it, if anything?
there are a couple of things that might explain it. I mean, if you go to microbiologists and they see this, they're like, firmicutes have outer membrane proteins. They're not supposed to have it. They're like, do they really have an outer membrane? And so then some people call them negative acutes, which because they have like an atypical outer membrane. Do I have any microbiologists in the crowd? Okay. Oh. So then you won't be able to sign up for yourselves. But So the things with fewer are oftentimes weirder, less studied things, which means that maybe when we were training our initial, is it a barrel, right? We were training on proteins that you could express in E. coli because fundamentally we started with this set of structures which were beta barrels, and we knew they were beta barrels, and then we did contact maps. So that's where we started. And so when we said like, okay, how do we get a good MCC? That was based on those. So one possibility is we're missing many of those because they have, let's say, a smaller membrane, and having a smaller membrane would mean that their hairpins are smaller, and we're looking for larger hairpins, and then we miss them. So that's one possibility. Another possibility is that they have, you know, other types of proteins in their membrane, or they have a more permeable outer membrane, which ultimately means that then those types of functions would need to get done by the inner membrane. One thing that I can say that I think is particularly interesting is how many Bacteroidetes proteins we do pick up, given that there were no Bacteroidetes structures at the time. And Bacteroidetes are these organisms that often live in the gut and are sort of seem to have a lot of uptake needs. And so maybe the diversification of that type of uptake explains that.
If you replot this by fraction of the proteome, would it level out, or is it
No.
still the same?
No, that's in the paper, but I don't have it right here, but it's the same. We get to Bacteroidetes has sort of between 2 and 3% of the proteome. Oh, Chris.
Yeah, I'll ask the last one if there's no others then. Just, you know, also just fascinated by the unannotated new family members, and just wondering about like what the predictive structures of those might look like, if you glean any insight into what those
I
functions
mean,
might
when
be.
we look at alpha folds, they're barrels. So they're barrels, and if you look, I forget, you know, there's sort of an indication of, and we're doing actually strand number counts now. I have a really lovely undergrad who's working on it, Tasia. But you can see, right, they sort of, the smaller groups, I guess. So what's cool is that domain of unknown function 1302 has big amount of diversification. So that I think is like a very right place for looking. Whereas if you look at a hypothetical type 2, right, that's probably is all like the same strand number.
Is there any like maybe looking stuff like
Oh,
maybe
good
like
question.
Easons have some extra bits on there
Right.
that kind of give you like a little bit of, you know, hints as to what they might be.
Yeah, I don't remember, but I didn't look closely to that. What's this? Thank you.
Thanks for dealing with all the technical.
Yeah, I'm sorry. I think maybe you needed.
And
It is a school of technology.
Yeah.
So everyone knows we'll be back
They'll just-
here again next month. Normally we alternate, but because we had 2 months in a row at HMS, we're gonna do 2 months in a row at MIT. So back in this room and October.
Nice. Thank you all for coming.